We were yesterday talking about reversible inhibition, that is those inhibitor molecules which reduce the rate of an enzymatic reaction by reversible interaction with the enzyme molecule, and after the inhibition is over they can be physically separated and the enzyme can be recovered in its original catalytic functional form. Now, these reversible inhibitors are often categorized into three major classes based on certain general features, and some of the general features which are used to classify them are: one is pattern of binding of the inhibitor to either the enzyme molecule or the enzyme substrate complex, or in other words it leads to a specific mechanistic steps which are involved in the inhibitory effect. That means, the enzyme reaction rate is modified, reduced rather, to a new state in the presence of those inhibitor molecules. Based on that mechanism, then a rate expression is evolved, which also has characteristic features and for a particular class of inhibitor. Then one looks at the effect of the inhibitor on the Michaelis-Menten constant and the maximum reaction velocity of the reaction, and a particular class of inhibitor gives you a typical effect on the value of Km or Vm. Then another feature which can be looked at is the effect on the double reciprocal plot, the Lineweaver plot, and the nature of the Lineweaver plot, which is influenced as a function of inhibitor concentration, will also tell you about the class of a particular inhibitor. Then I think finally the type of inhibitor also influences the ultimate degree of inhibition, and as I mentioned earlier, the degree of inhibition is defined as or rather I is equal to V naught minus V I upon V naught, where I is the degree of inhibition and V naught and V I are the enzyme reaction rate in at concentration of inhibitor at 0 and at some particular concentration I for the enzyme reaction. Now, basically any inhibition pattern is studied on these basic lines, and yesterday we had looked at competitive inhibition, and to summarize the nature of competitive inhibition, the basic mechanistic step was that the inhibitor combines with the enzyme in a manner that prevents substrate binding. It may occupy the same binding site, it could also interfere with the binding due to steric hindrances in such a way that the binding of the substrate is prevented fully or partially, and essentially substrate and the inhibitor are mutually exclusive. Now, then their effect on the kinetic parameters of the enzyme reaction are just to increase the apparent Michaelis-Menten constant. The maximum reaction velocity remains unchanged. Then the inhibition constant, that means the reversible binding of enzyme with the inhibitor and the corresponding dissociation constant, is equivalent to the concentration of inhibitor that doubles the slope of initial reaction velocity inverse double reciprocal plot, that means 1 upon V versus 1 upon S plot. Now, if you recall, in the case of competitive inhibition, we had seen that your 1 upon Vi was equal to Km upon Vm 1 plus I over Ki into 1 upon S plus 1 upon Vm. The Michaelis-Menten constant is modified by the amount of this much 1 plus I over Ki, and therefore, the value of Ki essentially is equivalent to that concentration of the inhibitor at which the slope of the double reciprocal plot is double. That means, from the initial slope the value of the slope doubles, and that concentration of inhibitor gives you the magnitude of Ki. Then of course, degree of inhibition for the Michaelis-Menten for the competitive inhibition also depends upon all the parameters like, and the expression for degree of inhibition for competitive inhibitor can be written as Km into I over Ki whole divided by Km 1 plus I over K I plus S. So, basically as you will notice that the degree of inhibition depends on substrate concentration, it depends on the inhibitor concentration, it depends also on Km and Ki. All the parameter influence the degree of inhibition at any given inhibitor concentration for the competitive inhibitor. This is what we had discussed yesterday, and now if we come to the next class of major class of reversible inhibitor is a non-competitive inhibitor. Now, the classical feature of a non-competitive inhibitor in terms of binding with the enzyme or this enzyme substrate is that it in the inhibitor does not influence in any way the substrate binding. Essentially, substrate as well as the inhibitor bind reversibly, randomly and independently at different sites. That means, the binding site on the enzyme molecule for substrate are different than the binding site on the enzyme for the inhibitor molecule, and therefore, the molecules and the enzyme molecules that have bound to both enzyme, both substrate as well as inhibitor, and therefore, have the ESI complex, they are functionally inactive. And therefore, one can look at the reaction mechanism as E plus S goes to ES goes to E plus P, the classical root of your let us say K1, K minus 1 and K2. The enzyme can also bind to inhibitor, giving you EI, and this EI can also, because this the binding site for substrate is different, this can again bind to form ESI, and this ES can also bind to I to give you ESI. Basically, the enzyme substrate and inhibitor complex, the triple complex can be arrived at either from this route or from this route. There is no problem, because the binding of substrate and inhibitor do not interfere with each other. They can bind reversibly, randomly and independently. There is no interaction with them, and but only thing is that this ESI complex once formed is catalytically inactive and does not lead to product formation. It is only ES complex which will lead to product formation. And of course, in a simplistic this mechanism, we notice that we have a dissociation constant Km for the enzyme substrate binding and dissociation constant Ki for the inhibitor binding. And also we assume that whether the enzyme substrate complex bind to inhibitor or the free enzyme binds to inhibitor, the dissociation constant is identical Ki. Similarly, here when substrate binds to EI complex is also Km. So, as I mentioned earlier that there is no interference by binding of substrate or inhibitor onto the enzyme in their subsequent binding process. And under such conditions, one can write the rate expression for a non-competitive inhibitor as S into Vm over Km 1 plus I over Ki plus S 1 plus I over Ki. Or just to simplify, to look at the effect on the Km and Vm values, if we divide both numerator and denominator by 1 plus I over Ki, S into Vm divided by 1 plus I over Ki whole divided by Km plus S. Now, so one can very clearly visualize that the effect of a non-competitive inhibitor is to influence the maximum reaction velocity, and the maximum reaction velocity is reduced by a factor of 1 plus I over Ki, whereas the Km value remains unchanged. There is no effect on the Km value. Now, if you compare in contrast to the competitive inhibitor, where the Km value was altered, here the Vm value get altered and the Km remains. The modified maximum reaction velocity is equal to Vm upon 1 plus I over Ki. Now, basically the if you look into mechanistic concept, it means essentially that the total fraction of the enzyme molecule which was originally present for catalytic function has now got distributed into ES, EI and EIS, and only a part of it which is in the form of ES is catalytically functional, and therefore, the maximum reaction velocity which is a function of enzyme concentration, and because this fraction is reduced. So, the net effect on the maximum reaction rate is experienced, but mind it that it does not influence the K2 value, the turnover number, because as we know that the Vm essentially is K2 into E naught. Now, here E naught corresponds for a in the presence of a non competitive inhibitor to that fraction of the total enzyme which is in the form of ES. The ESI complex form is functionally inactive, and therefore, the K2 is not altered, it is the E0 which is modified or reduced by a factor of 1 plus I over Ki, and therefore,
so how can the E0 be reduced? It is a fixed quantity.
No, E0 is a fixed quantity when there is a non-inhibited reaction. Once the inhibitor is present, now then E0 will be distributed into free enzyme, ES, EI and ESI. So, basically E0 is not reduced, but the effective concentration of the enzyme, effective total concentration of enzyme, I can put it real, let us say E naught, E bar, E prime not actually, the effective concentration of the enzyme which is catalytically functional is reduced, and it is only the ES component which is catalytically functional.
So, why is ESI inactive? I mean, if they are independent sites, totally different sites.
Yes, in many cases, when the binding of substrate can influence the catalytic function on the substrate binding, and
they are independent.
But as binding site, the binding wise it may be independent. You see, there are two parts in the active site. One is the binding process, which is not influenced, which is elaborated by our dissociation constant. The binding part of the active site is demonstrated by the dissociation constant. The second part which is demonstrated is the catalytic function, which is given by K2 or the turnover number. Now, that remains, that can be influenced by the binding of the inhibitor.
So, but here you are saying K2 is constant and the turnover number is unaffected.
Turnover number is unaffected here in the sense that yes, in the case of the non-competitive inhibition, the turnover number will be unaffected as far as the kinetic parameter is concerned, but it can influence the catalytic capacity of the molecule, the enzyme molecule in total.
How do we physically picturize it? Because I mean, we have an enzyme, we have two different sites, one of which enzyme is substrate is bound to the other, the inhibitor. I mean, if they are independent, I don't see a reason why they should be inactive. Yes, even though I is attached to it, why should it be inactive? So,
no, this is where the question comes. When the enzyme and when the substrate and the inhibitor both are bound, it influences the whole molecule in such a way that the catalytic function is lost. It is a dead end complex, and the substrate cannot then the enzyme substrate part, the binding site on the enzyme which is bound by the substrate is not able to give the product. But when I say K2, I am referring K2 in the sense of the original catalytic function which was present, which is retained, and only in the presence of the bound inhibitor on the enzyme site, on the inhibitor binding site, the catalytic function is lost. The double reciprocal plot of the in the presence of non-competitive inhibitor is given as Km over Vm, 1 upon V is equal to Km over Vm, 1 plus I over Ki into 1 upon S plus 1 upon Vm, 1 plus I over Ki. Now, it clearly indicates that in the case of a non-competitive inhibitor, both the slope as well as intercept will undergo a change, and both the slope and the intercept are increased by a magnitude 1 plus I over k I. And therefore, if you look at the pattern of the Lineweaver plot, you essentially get at a increasing concentration of I, you get, let us assume that this is I is equal to 0 and some values of I as I1 and I2, which is increasing. The slope and the intercept are both increasing by a magnitude of 1 plus I over k I, and therefore, one can determine the value of k I as we saw in the case of competitive inhibitor by a secondary plot. That means, either you determine slope or intercept at different inhibitor concentration and make a plot between I and slope or intercept, you can get a straight line, because slope will be equal to Km over Vm 1 plus I over Ki, and therefore, one can determine the value magnitude of Ki either by plotting the slope versus the inhibitor concentration or by intercept. Either way one can determine, and that can give you the magnitude of inhibition constant. The third major category of the reversible inhibitor are uncompetitive inhibition. Now, in this category of reversible inhibitors, the inhibitor binds reversibly to the enzyme substrate complex. It does not have any mechanism to recognize the enzyme free enzyme. Only when the substrate is bound to the enzyme, it can bind to the enzyme substrate complex, and after binding of the inhibitor, the ES complex becomes a inactive complex, and the inhibitor does not bind to the free enzyme. So, essentially one can see ES E plus P, this can bind to inhibitor giving you ESI. Now, this is the dissolution constant Ki, classical Km and K2. So, essentially the inhibitor can bind only to the ES complex and does not bind to the free enzyme, and therefore, it is only responsible for the site. Now, such situations are very rare as far as the uni-substrate reactions are concerned. Very few cases, almost rare cases are there where one notices this kind of uncompetitive inhibition. In most of these cases are usually in relation to bisubstrate reactions, particularly because you notice here that the inhibitor binds after the substrate is bound. So, there is a sequence of binding. So, I mean, in cases only where there is a ordered sequence of binding of the ligands, then only the uncompetitive inhibition is feasible. In cases where the binding is random, that means the inhibitor and substrate can bind randomly without any sequence, the mechanism of uncompetitive inhibition is very rare, and therefore, but still the idea of uncompetitive inhibition has been very useful in understanding the reaction mechanism for bisubstrate reactions. And essentially, when we look at bisubstrate reaction, we will notice that this inhibition patterns gives you a lot of information about the sequence of binding of the substrate in the case of a bisubstrate reaction. Now, in the case of a uncompetitive inhibition, the reaction velocity can be written as S into Vm upon Km plus S 1 plus I over Ki. Again, if you just to bring it in the same format of Michaelis-Menten equation, if you can divide both numerator and denominator by your 1 plus I over Ki whole upon Km upon 1 plus I over Ki plus S, you notice that both Km as well as Vm, you have K dash m is equal to Km upon 1 plus I over Ki. You have V dash m is equal to Vm upon 1 plus I over Ki. Both the parameters are reduced by a factor of 1 plus I over Ki, and the double reciprocal plot of in the presence of an uncompetitive inhibitor will be Km over Vm into 1 upon S plus 1 upon Vm into 1 plus I over Ki. Therefore, you will notice that the uncompetitive inhibitor does not change the slope remains unaltered, but the intercept is increased by a factor of 1 plus. Now, this is your intercept, and the intercept 1 upon Vm on the y axis is increased by a factor of 1 plus I over Ki. And therefore, if you look at the typical pattern of a uncompetitive inhibitor is something like parallel lines, where both slope and where the slope does not change, but the intercept value 1 upon Vm is changed, increased by a factor of 1 plus I over Ki, and a secondary plot of 1 upon K dash m versus I. Now, if you just take it, let us say 1 upon K dash m will be equal to 1 upon Km plus I over Km into Ki, and if you make a secondary plot, you will get the value of Ki. So, in these all these cases, therefore, by following a typical pattern, that is by understanding the reaction mechanism, the binding of the substrate, binding of the inhibitor and substrate to the enzyme, their interaction between each other, one can really arrive at a rate expression. One can also look at the double reciprocal equation, Lineweaver equation, make a double reciprocal plot, see the nature of double reciprocal plot, and can identify that what is the nature of the inhibitor which we are talking about by understanding these. In many cases, we will also notice that the actual inhibitor does not follow a very typical pattern, and not surprising that very often, let us say, we end up a profile something like this, which intercepts, which does not intersect on the y axis nor on the x axis, and it intersect at somewhere on the middle point. Now, such a situation can be considered something away from the ideality. There are kind of mixed type of inhibition, and as a matter of fact, one of the most commonly observed mixed type of inhibition is what we call as linear mixed type inhibition. Now, this is a typical case of a non-competitive inhibition. If you recall non-competitive inhibition, where we had where we have considered the binding of the inhibitor to either the substrate or the free enzyme freely. Now, in that case, we have assumed one basic factor, that the dissociation constant for binding of the inhibitor to the enzyme and to the binding of the inhibitor to the enzyme substrate complex is identical. But in many cases, this may not be so. The dissociation constant for binding of inhibitor to enzyme may be different than the dissociation constant for inhibitor binding to ES complex, and there might be a let us say a interaction factor of alpha, and if we consider that, in such a case we ES going to E plus P Now, E plus I, it binds with a dissociation factor of K I, E I. Now, E S binds with inhibition constant of alpha K I to E S I, and here also substrate binds. Now, obviously, if the inhibition constant for this is K m, and simply by if the system has to come to an equilibrium, then the equilibrium constant for reaching E S I from either side must be identical, and that way this inhibitor constant will also become alpha K m. And therefore, in most cases it so happens that the inhibition constant for binding of inhibitor to the free enzyme or binding of inhibitor to the enzyme substrate complex are not identical as assumed in the non-competitive inhibition, and therefore, they influence the presence of the inhibitor on the enzyme changes K m also by the same factor alpha K m, and in such cases one can the rate expression becomes modified as V m into S upon K m 1 plus I over K I plus S into 1 plus I over K alpha K I. And 1 upon V I is your K m over V m into 1 plus I over K I upon S plus 1 plus I over alpha K I upon V m. And obviously, for cases where alpha is greater than 1, you will get a profile as I showed earlier, that is your the enzyme will the double reciprocal plot of 1 upon S versus 1 upon V at different inhibitor concentrations will intersect at a point on the left hand side of the y axis. And if you consider the slopes and the intercepts of this plot, you can get this a replot of the slope will give you K I, and if you plot if you replot the intercepts, modified intercept as a function of inhibitor concentration, it will give you alpha K I, and therefore the value of alpha can be determined. That means the magnitude by which the dissociation constant of binding of inhibitor is modified can be determined, and essentially such profiles are indicative of modification of the dissociation constant as a function of substrate binding in the non-competitive, and such cases are very commonly found, and in most of the practical cases they are usually not the ideal type of inhibition patterns, they are rather mixed type of inhibition patterns, and these mixed inhibition patterns are essentially attributed to modification in the dissociation constant as they are obtained in the. Now, another feature which you recall that I mentioned, a general feature which gives you the properties or the characteristic of any type of inhibition pattern is degree of inhibition. If we just recall from all our rate expressions that we wrote about the different types of inhibition pattern, I as equal to V naught minus V I upon V naught. Now, you will notice that for non-competitive, if you substitute the rate expression for the inhibited and the uninhibited reaction, for the non-competitive the value of I will be I upon K I plus I, which indicates that the degree of inhibition is independent of substrate concentration. Whatever the substrate, unlike in the case of competitive inhibition, where increasing substrate inhibition can significantly decrease the degree of inhibition, that means that the extent by which the rate of the enzyme catalyzed reaction is reduced by inhibitor can be reduced by increasing the substrate concentration in relation to the inhibitor concentration. In the case of non-competitive inhibition, whatever is the concentration of substrate, it does not influence the degree of inhibition, and it is totally dependent on the concentration of inhibitor and the Ki. In the case of uncompetitive inhibitor, the degree of inhibition is S into I upon Ki into Km plus S and 1 plus I over Ki. Now, which the degree of inhibition will rise from, let us say, S equal to 0 when there is no reaction, to a limiting value of I 1 plus I over Ki at very high value of substrate concentration. When substrate concentrations become very high, the limiting the degree of inhibition will be 1 plus I over Ki, but it will depend, like competitive inhibition, on the substrate concentration, inhibitor concentration, Ki and Km, all the parameters. Now, in the case of linear mixed inhibition, essentially the degree of inhibition will be largely dependent upon the magnitude of alpha, the magnitude by which, like in the case as I mentioned earlier, that the mixed inhibition is a special case of non-competitive inhibition, where otherwise substrate concentration does not influence the degree of inhibition, because the binding sites are different and they do not interfere with each other. But in the case of mixed inhibition, the magnitude of inhibition will be dependent on the ratio of the dissociation constant for free enzyme and enzyme substrate complex with the inhibitor, and the alpha will determine the magnitude of the degree of inhibition. So, these are some of the typical characteristic categories of the inhibition patterns which are commonly noted, and as I mentioned in the beginning, that they have a key role to play in understanding the mechanism of a variety of complex reactions, particularly the bisubstrate reactions. The inhibition patterns obtained for bisubstrate reaction gives you a lot of information about the binding pattern of different substrate or the sequence of binding of the different substrate in the case of a multi substrate reaction. While in the case of a single substrate reaction, that is what we have been talking so far, the life is simple, because there is only one substrate and we are concerned with the binding of that substrate. In the case of a bisubstrate reaction, we have much more possibilities, much more types of mechanistic approaches are feasible and they are taken into account.
So in the case of uncompetitive inhibition at very high substrate concentrations, what will be the value of degree of inhibition?
At very high substrate concentration, that when we say very high substrate concentration, we are talking as is much greater than Km,
Yes,
and we are talking in the zero order regime,
yes,
and then the degree of inhibition at very high substrate concentration will be significantly high, because then it will reach the limiting value of 1 plus I over Ki.
How is it reaching that limiting value, sir?
Huh? You see, this becomes, then Km becomes K and S gets cancelled,
Yes, sir.
and then you get degree of inhibition will be I upon Ki into 1 plus I upon Ki.
Yes.
It reaches the limiting value of 1 plus I upon Ki maximum, but otherwise at low substrate concentration this will then be somewhere in between 0 and that value. Now, another way to look at this which I like you to understand and appreciate is to look at the inhibition pattern is in a generalized way rather than in specific cases. And you will notice that if we look at a very generalized scheme of inhibition patterns, if we look at, let us say, enzyme going to enzyme substrate complex going to E plus P, then enzyme also enacting to EI, ESI and obviously ESI. Now, if we consider each of the reversible steps, of course, we are not here concerned with the irreversible step, because that determines the rate of the reaction. The interaction of the reversible steps, if we say that, let us say the dissociation constant for E is this step is ES and this is KEI, this is KESI, this is also KES. Now, in the three different cases that we talked about for the type of inhibition, in the case of a competitive inhibition, in the kind of a competitive inhibition, your magnitude of what will be the magnitudes of the respective your KESI will tend to infinity. See, KES and KEI will have definite magnitude. That means the enzyme can bind either to inhibitor or to a substrate, and KES and KEI will have definite magnitude, but the KESI either will be infinity, and that is a typical case of a competitive inhibitor. In the case of a non-competitive inhibitor, your KESI will be equal to KEI. That means these two dissociation constants are identical, and that is what, as a matter of fact, in the case of a mixed inhibition they are different by a factor alpha, and in the case of a uncompetitive inhibition the magnitude of KEI is infinity, or rather tends to infinity. That means the inhibitor cannot bind to the enzyme. So, basically I mean the three cases are typical example of three extreme situations of the inhibitor binding. In the competitive inhibitor, the ESI, the dissociation or the binding of the inhibitor to ES complex is not feasible, and the dissociation constant is infinity. In the case of non-competitive inhibition, the two, it can bind either to E or ES, and the dissociation constant is identical. In the case of a mixed inhibition, they are not identical, but they have a magnitude and both are related by a ratio of alpha, and which determines essentially the degree of inhibition. In the case of uncompetitive inhibition, the binding of inhibitor to the free enzyme, the corresponding dissociation constant is again approaches to infinity. That means the binding to free inhibitor is not feasible. Now, with that view, I think we would like to look at the other the bisubstrate reactions subsequently, and we will stop at this point.